Good morning. My name is Miss Beard, and I'm just going to talk about DNA, and that kind of is a reoccurring theme in a lot of our science classes. DNA stands for deoxyribonucleic acid, and it's found in the nucleus of living organisms. We've got plant and animal cells. It's also found in bacteria and prokaryotic cells, just not in that nuclear membrane. Let's just watch part of this video here. Wanted to mute that. All right, so this is going to zoom in. We're going to see the DNA is in this picture coiled up in something we call chromosomes, and we have many of these. And if we were to stretch the DNA out linearly from this chromosome, it'd be about like 2 meters long, okay, from end to end. So on these chromosomes, they're coiled around these histones, and there's sections of chromosomes called a gene, right? Genes are unique because they code for proteins, and proteins make up structurally a lot of your body. If you've been in the biology class, you've learned that nucleic acids, DNA being one of those, is one of the 4 main macromolecules. So its function is we open up and unlock these genes to code to make all kind of things from head to toe. Your hair to insulin proteins, hemoglobin, transport proteins, actin, myosin, bone, all of it, right, is coded in these genes that are found on these chromosomes. You see here these little colored things that look like rungs on a ladder. So DNA's shape is a double helix, and what we find in the center of that are our base pairs, right? We have base pairing rules. And a lot of the structure for DNA was founded by Rosalind Franklin's X-ray, where she figured out the shape, and then Watson and Crick helped distinguish what these rules are that the base pairs follow. I'm going to pause the video and switch to this one. So molecularly, in this picture, we see one of our bases. We have 4 nitrogenous bases: thymine, which you see as a T represented in this picture, adenine, guanine, and cytosine. What I'm attaching here in that image was a sugar, all right, the deoxyribose, and then this piece here, which is your phosphate group. So the nitrogenous base, which can vary, and you see it as different colors here in this video, adenine, thymine, guanine, or cytosine, is attached to a 5-ring sugar called deoxyribose, where it's missing a hydroxyl group, and then this piece, this yellow being the phosphorus, a phosphate group on the end, and we call this a nucleotide. Nucleotides are repeating units that make up the whole big structure. Okay, so let's watch this part a little bit more. I'm going to show how we would connect nucleotide to nucleotide. So following base pairing rules, adenine, which is here, will always bind with thymine. And that's universal throughout all living organisms. All right, adenine is always going to bind with thymine. One of these, the adenine and the guanine, are called a purine. That might not be relevant to your course, but it just means that there are 2 rings. So molecularly, it's a little bit wider, it's a little bit bigger in terms of the helix shape, all right? And then thymine and cytosine are called pyrimidines, and they have a single ring shape, all right? Purines, A and G, pyrimidines, C and T. So let's watch this part. Those join together in the center of the helix. Now, in that picture, they're connecting by magnets, but they actually bond by hydrogen bonding. Hydrogen bonding is one of the weaker types of bonds, and it allows me to unzip or open up the DNA like you see back in this video. And the reason I need to open up or unzip the DNA is so that I have access to those genes so I can make proteins, okay? So hydrogen bonds are not super strong, but they keep the helix connected between those base pairs in the center. Let's look at the other base pairing rules. I mentioned C for cytosine, G for guanine. What might be slightly different about this pairing is they are connected by 3 hydrogen bonds versus the 2 hydrogen bonds that you see in adenine and thymine. And so these connect, and I attempt to show you molecular 3D shape, this double helix when we connect two nucleotides together and then they stack. What you see along the exterior are the sugar and phosphates, and on the inside you see the two nitrogenous bases that are joined together. Let me hop back over to this video to show what the point of DNA is. So this might be a lesson that comes a little bit later, but you have an enzyme that unlocks or unzips DNA at that gene state, and then another enzyme comes and builds this piece of messenger RNA. So one thing you see different is this U. U stands for uracil. mRNA is single-stranded, RNA is single-stranded, and it's basically the same structure as DNA. The nucleotides look similar, except for you'll never see any thymine in messenger or any types of RNA, right? It's replaced by a different nitrogenous base called uracil. So this uses the basic rules, right? Base pairing rules. A's bond with U's instead of T's. C's are still with G's. And this copies the length of the gene that is needed, right? So I go for as long as the gene. This peels off and it's going to leave the nucleus through the nuclear pores. And I might speed it up a little bit. And it's going to find its way out into the cytoplasm of the cell, and it's going to conjoin with a ribosome. Ribosome is where I read this messenger RNA and I bring in amino acids in a unique order and build a protein from it. Okay, so this is kind of that process sped up, and then amino acids are this little growing ball of chains up here. Amino acids are the building blocks of proteins, and those have a unique fold and make a unique molecular shape, all right, that it's supposed to be. Proteins are very lock and key, meaning that their shape is crucial for their functioning in the human body. And so if we don't have the correctly shaped protein, then it won't work correct in the body. So that goes a little overview of DNA, its purpose, what does it do. DNA, you could look a little deeper and connect it to forensics. DNA is unique to each person. If you are an identical twin, then your DNA is an exact match, but otherwise your DNA is variable to you and unique to yourself. And so they use that DNA to match up. It's called DNA fingerprinting. It's not actually your fingerprint, but it's a broken up kind of gel electrophoresis like type of deal where I see a banding pattern that's unique to me or to you or to anybody, and then if they have trace evidence that they've collected at the crime scene that's also got DNA in it, what they do is like a match, and that's DNA fingerprinting. So that's used in forensics. We study the human genome. It's used in gene therapy, so there's a lot of diverse and really cool things about DNA. Hope this helped you understand the structure a little bit more, and if you ever need any help, just let me know. Have a great day.